In this video, we are going to discuss about cell envelope of bacterial cell. The bacterial cell envelope consists of three parts. I have already mentioned the names of three parts: glycocalyx, cell wall, cell membrane. Cell membrane is also known as plasma membrane. Let us see glycocalyx. Glycocalyx lies outside the cell and it is made up of protein and lipid, or you can say it is made up of glycolipid and glycoprotein. The innermost part is protein rich or peptide rich, it is glycoprotein, and outer one is glycolipid or carbohydrate rich, it is also known as mucilage. So this glycocalyx is not very essential for the survival of cell, so it is known to be secondary, having a secondary function. Secondary functions like protection against desiccation, resistance, from toxins and chemicals, now this glycocalyx are of two types. If it is loosely bounded to the cell wall, it is known as slime layer, and if it is thick and bound tightly to the cell wall, it is known as capsule. Now after this, here in this diagram we can see this part is glycocalyx, the outermost one is glycocalyx, okay, this one, okay. So this is a glycocalyx, and then we can see the next membrane, that is your cell wall. Cell wall, there is a gap between cell wall and plasma membrane. This gap or this space is known as periplasmic space. Let me write over here. Okay. So this is periplasmic space. The space between cell wall and plasma membrane is known as periplasmic space. The cell wall also consists of two parts. The outer membrane, now you can see outside this black color boundary, this blue color I have shown here, this is outer membrane, okay, and inner one is peptidoglycan. So peptidoglycan, in my previous video I have also explained peptidoglycan. Peptidoglycan is a network of NAG and NAM, that is your N-acetyl glucosamine and N-acetyl muramic acid. N-acetyl glucosamine, which is your NAG, and N-acetyl muramic acid. So now this peptidoglycan layer varies according to bacteria. If the bacteria is gram-negative bacteria, then peptidoglycan layer will be very thin and it will be about 10 to 20% in gram-negative bacteria. But if bacteria is gram-positive, then the peptidoglycan is about 70 to 80 nanometer thick. So now our cell wall, it is made up of peptidoglycan and outer membrane. Outer membrane mainly consists of protein and lipid. So this outer membrane is it contains mainly lipid in gram negative. In gram negative bacteria, then about 80% lipid. It is made up of 80% lipid. So we can see we have seen in that gram staining also that outer membrane got dissolved because of acetone. So here in gram negative bacteria, 80% of lipid is present in outer membrane, and in case of gram positive, there is a thick peptidoglycan and outer membrane is quite thin, or there is no outer membrane, there is a peptidoglycan, wide peptidoglycan in gram positive bacteria. So cell wall mainly consists of this two: protein, lipid and carbohydrate. Next we are going to see plasma membrane. Plasma membrane or cell membrane. So this cell membrane or plasma membrane, it is lipid bilayer. Let me write over here. Plasma membrane is lipid bilayer. Okay, we can see over here there is a phospholipid polar molecule having two fatty acid chain, and in the same way so this kind of lipid bilayer is present. It is the innermost part of cell envelope. So cell envelope, bacterial cell envelope generally consists of three parts, that is glycocalyx, cell wall and then cell membrane. Glycocalyx is the outermost one, then cell wall and then cell membrane. Cell membrane is also known as plasma membrane, and plasma membrane is semi-permeable membrane which allow only some of the smaller molecule to move outside. Okay, thank you.